Hello, everyone, and welcome to today's webinar, Pgta Simplified, which of the leading NGS solutions is easiest to implement? I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com/rh. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Adam Goodman, the Director of Pre-Implantation Genetics at NextGen MDX. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Adam, you may now begin your presentation. Welcome.
Thank you for the introduction. My name is Adam, and I run the Pgta department for NextGen MDX, a U.S.-based genetics lab located in Grand Rapids, Michigan. Whether you're expecting or one day hope to be, genetic screening can help you make informed decisions about how to move forward. At NextGen MDX, we make it our mission to ensure our patients have access to best-in-class science, unrivaled accuracy, and personal genetic counseling designed to help them chart a course for their family's future. To that end, we offer our clients a host of best-in-class services, including state-of-the-art carrier screening, pre-implantation genetic testing, non-invasive pregnancy testing, hereditary cancer screening, molecular imaginosis testing, and more recently, we have become the largest COVID-19 testing laboratory in the state of Michigan, having run over 300,000 tests since March. Today, we'll be focusing on Pgt and a technical research study our team performed comparing two of the leading NGS-based Pgta assays. I have been working in the Pgt field for over 10 years now. When I first started out, we were still using FISH and PCR-based tests to assess chromosomal composition in day 3 embryo biopsies. And I've been able to experience firsthand the evolution of Pgt through many different platforms. Today, we will be discussing two of the world's most widely utilized and trusted assays. The longtime champion of high-resolution Pgta, VeriSeq PGS, originally designed by Illumina and now owned by Vitrolife, and the more recent newcomer, Retrofit PGS by Thermo Fisher Scientific. As the head of pre-implantation genetics for NextGen, a key part of my job is to always be on the lookout for new technologies to advance our Pgt program, and for almost a decade I have been using VeriSeq. I still believe it is a good product, however I am always open to new ideas, and when the latest version of Thermo's Retrofit assay came along, it proposed to offer the same level of higher resolution sequencing as VeriSeq, but with an easier protocol and lower price. Since at NextGen we employ both Illumina and Thermo Fisher sequencers on a large scale in our other divisions, we are fortunate in that we can easily switch between sequencing platforms for Pgta, and we decided to give the Retrofit assay a try to see if it would meet our high standards. In the past, having spent many years behind the bench as a lab technician, and now on the directorial side of things, I am well versed in evaluating new Pgt products. As such, I ran the Retrofit assay through its paces, and afterwards I was asked by Thermo Fisher if I would be willing to share my experience. Before I get to the experiments, I want to talk about what I look for in a Pgta assay. The goal of Pgta as a science is to identify those embryos with the greatest potential of resulting in a healthy pregnancy and baby. To best accomplish this goal, we need an assay that has a resolution high enough to accurately detect not just whole chromosomal gains and losses, but also smaller segmental deletions and duplications, as research studies have shown that they too can be responsible for reduced implantation and increased miscarriage rates. At the same time, we must be sure that the aneuploidy events that we are detecting really exist. If an assay does not provide tools to properly investigate sample and data quality, it will be prone to false positives. And finally, cost. I would be lying if I told you cost is not a big factor. The Pgta field has become extremely competitive over the last decade. Many of our IVF lab clients are big believers in improving their success rates with PGT, and lower cost means greater utilization. As such, without overwhelming evidence of superior technology, even modest price increases are not going to be accepted in this field. Here are some of the key aspects of any prospective Pgta assay that need evaluation. Resolution and accuracy. I want to know the assay provides true 24-chromosome high-resolution data with the ability to reliably identify mosaic and segmental aneuploidies. Reliability and precision. Running isolated genomic DNA through a Pgta assay is easy. You're going to get great quality data every time because you have a pristine quality sample. I am more interested in testing out real-world analogs of variable quality because in practice, not all samples received by our lab are going to be perfect. Data interpretation. Does the assay provide strong software capable of allowing our lab to thoroughly investigate samples, not just for aneuploidies, but also for assessing the quality of the data itself, so that we can better separate sample noise and amplification artifacts from true aberrations? This is a challenging task, and as far as I know, NextGen is the only company willing to provide Pgta data analysis and reporting services separate from the lab work side of things. Throughput and ease of use. How long does the assay take to run? How much of that time is hands-on, and how amenable is the assay to automation? And finally, I want to know more about the associated costs than just the per sample price under ideal circumstances. I want to know what samples per run size options are available and the total per run cost associated with each. Very few labs have enough sample volume to fill every run to its maximum. You need to know what your costs will be for runs performed on a 100% capacity. Labor costs are also another important factor. What level of experience is required and for how long at the various stages of sample handling. Assays that use more automation can free up senior technician time without reducing quality. Additionally, there can be significant differences in ancillary costs such as plastic consumables and safety equipment. A note on Pgta assay resolution. Yes, it is possible to dramatically lower the cost of a Pgta assay by turning down the resolution. Whether you're using an Illumina flow cell or Thermos ion sequencing chip, each has a limited amount of physical space available that can be utilized to gather sequence data from samples. It is simple math. Lower resolution means more samples fit on a sequencing chip, and the more you can cram onto a chip, the lower your cost per sample. Doubling the number of samples run on a chip halves your cost per sample, but it also halves the amount of sequencing data that can be gathered from each sample, and this can seriously inhibit your ability to detect smaller deletions and duplications. So how exactly does this happen? Changes in chromosomal copy number do not have to occur as whole chromosome gains and losses. As such, a higher resolution assay is inherently better at detecting partial aneuploidy events because you cannot detect deletions and duplications in areas where you have no coverage. Looking at the example on this slide, imagine one is sequencing only the gray regions of this chromosome. But what if our deletion occurs in the green region here outlined by the yellow box? Because the assay is only looking in the gray regions, it would not detect the loss, and a failed implantation or miscarriage is the most likely outcome. Such partial aneuploidy events can occur randomly, or in the case of translocations, they can be driven towards specific locations with high frequency. This is a big deal because studies have put the percentages of Pgta embryos with segmental aneuploidies at around 8%. That means if your assay cannot see these, your misdiagnosis rate could be as high as 1 in 12. Yes, lowering the resolution can lower the cost of the test, but it's simply not worth the risk. For our investigation of resolution, we purchased 10 different commercially available cell lines with known abnormalities ranging in size from whole chromosome down to 1.5 megabases. One important distinction here is that we tested actual intact cells, not isolated DNA, as is used in most assay manufacturer validation studies. I believe that this approach provides a better analog of real-world performance with regards to embryo biopsies. We diluted cell suspensions down to the amount found in typical Pgt biopsies, aliquoting approximately 5 to 10 cells per collection tube. We then ran them multiple times through both the VeriSeq and ReproSeq platforms to see if we could empirically find any difference in their limit of detection with regards to the size of segmental deletions and duplications. What we found was both assays outperformed their manufacturer's stated stock resolutions of 10 megabase pairs, reliably detecting segmental changes down to about 5 megabases before we started to see any missed calls. Both assays showed identical results for all samples, not just in the limits of detection, but all the corresponding copy number charts were virtual overlays of each other. Here are two example copy number variation plots from the experiment. Chromosome numbers listed across the x-axis and copy numbers listed across the y. I realize the numbers are hard to see, but basically what one is looking for is movement away from a baseline of 2 copies per chromosome on the y-axis. With these samples, there are only 2 areas that show such aberrations. All the way to the right, you can see a loss in copy number of chromosome Y, which is expected given this is a female sample. And a little to the left of that, our copy number for chromosome 21 has jumped from 2 to 3, indicating trisom 21. What is particularly interesting about this sample is that both ReproSeq and VeriSeq saw a full gain of chromosome 21 when the cell line was previously karyotyped as having only a 33 megabase duplication on chromosome 21. Neither assay showed any indication of euploidy in any area of chromosome 21. The most likely explanation is that ReproSeq and VeriSeq have a higher resolution than the array-based assay used in the original karyotyping of the sample. However, as we tested actual intact cells and not isolated genomic DNA, it is also possible that the abnormality was unstable in the cell line culture. For our assessment of accuracy, we again wanted to test an analog more rigorous than isolated DNA. So we partnered with an IVF lab in Florida to retest embryos that were previously predicted to be aneuploid by a third party using a SNP-based array platform. Since the experiment involved running the sample simultaneously through both the ReproSeq and VeriSeq platforms, this was helpful in that it could potentially provide a tiebreaker in the event of a discrepancy between the two NGS-based assays. Additionally, we tested not just a trophectoderm biopsy from these samples, but also the remainder of the whole embryos themselves to assess concurrence between biopsy and embryo calls. After all, the whole point of Pgta is to be able to correctly predict the ploidy of the embryo. 22 embryos yielding 44 unique samples made it into the study, and NextGen was blind to the third-party aneuploidy predictions, which sample tubes contained biopsies, which contained whole embryos, and also to which embryo a given biopsy belonged. We found 100% concordance between ReproSeq and VeriSeq. At the fundamental level of calling a sample normal or abnormal, we also saw 100% concordance between the biopsies and their corresponding whole embryo sample. However, we did see 2 instances in which the abnormalities detected in the biopsies did not perfectly match that seen in the whole embryo sample. Those sample sets are seen here in red, and they are not necessarily surprising in that the abnormalities detected are related, even though they are not perfect matches. Other studies have shown that with mosaic samples, we sometimes see variations of the same abnormality when a sample is biopsied multiple times, which is a trisomy in one biopsy and a reciprocal monosomy in another biopsy from the same embryo. The more shocking discovery we made was that 4 of the embryos previously predicted to be abnormal by third-party SNP array were found to be euploid by NGS. In these instances, both the biopsy and the whole embryo samples were found to be euploid on both the ReproSeq and VeriSeq platforms. These samples are highlighted in red. There were 3 other discrepancies between the third-party testing and our 2 NGS-based assays seen in blue. However, in these instances, again, the abnormalities were related and likely implicate technical limitations of the SNP assay and or embryo mosaicism as the cause of the discordance. So how could nearly 20% of the embryos predicted to be aneuploid by a third-party SNP array turn out to be euploid on our 2 NGS-based platforms? Yes, it is theoretically possible that mosaicism could account for the difference. However, I find that scenario extremely unlikely. For this to happen, it would not only mean that nearly 20% of the embryos we tested were true mosaics, but it would require two fundamental conditions to be true on 4 separate occasions, as we saw 4 discrepant embryos. First, no euploid cells could have been a part of the original biopsy set for SNP array testing. This biopsy must have been composed of only aneuploid cells. And second, all or most aneuploid cells from this hypothetically mosaic embryo would have to have been excised from the embryo during the original biopsy, such that both the biopsy and the remainder of the embryo, which were sent to NextGen, could contain no aneuploid cells. To understand why this is extremely unlikely, we must first understand a little about the etiology of embryonic mosaicism. A mosaic embryo in this context is an embryo that contains a mixture of both aneuploid and euploid cells. Here we see 3 different types of mosaic embryos, with aneuploid cells represented in red and euploid cells represented in green. Aneuploid cells can be confined to the trophectoderm area, and as the trophectoderm goes on to form the placenta, this is known as confined placental mosaicism. Confined placental mosaicism can lead to a false negative if the biopsy is taken from an area which happens to contain no aneuploid or red cells. Embryos with generalized mosaicism have aneuploid cells spread throughout their structure. This type of mosaicism is less likely to result in misdiagnosis, as a given biopsy is likely to contain at least some aneuploid cells. If the aneuploid cells are confined to the inner cell mass, which goes on to form the fetus, it is known as fetal mosaicism. Because our study tested the remainder of the whole embryo in addition to a second biopsy, at least 80% of the aneuploid cells would need to be removed from the embryo during the original biopsies performed for SNP testing. Otherwise, the NGS-based platforms would show signs of abnormality, as 20% mosaicism is generally regarded as a limit of detection for high-resolution NGS-based Pgta assays. This effectively rules out 2 of the 3 types of embryonic mosaicism. First, embryologists are trained to not biopsy the inner cell mass of the embryo, as this is the region of the embryo that goes on to form the fetus. For this to be accidentally done on 2 separate occasions with 4 different embryos is thus highly unlikely. This rules out fetal mosaicism. Next, embryos with generalized mosaicism have aneuploid cells scattered randomly throughout the embryo, making it virtually impossible to perform a biopsy on the embryo and not leave at least some aneuploid cells behind that would later be detected during the testing of the whole embryo. This rules out generalized mosaicism, leaving us with just embryos in which aneuploid cells are confined to an isolated area of the trophectoderm. So assuming all 4 of these embryos had confined placental mosaicism, we would still need some very specific things to happen for the SNP assay to show aneuploidy, while the NGS assays showed euploidy. Take a closer look. During the first biopsy for SNP testing, we would need to see all or almost all of the aneuploid cells removed from the embryo, but with no euploid cells tagging along. Those aneuploid cells would then move on to undergo SNP-based Pgta, leading to the third-party aneuploid result. Next, the second biopsy, the one tested by our team, would need to be taken from an area of the embryo with no aneuploid cells. This biopsy then undergoes whole genome amplification, and since ReproSeq and VeriSeq use the same whole genome amplification chemistry, the WGA product can be split and sequenced separately on the 2 different platforms. If no aneuploid cells were contained in the second biopsy, both NGS assays would give a euploid result. Finally, and this is the most unlikely condition, if both biopsies occurred perfectly such that no aneuploid cells still resided anywhere in the remainder of the embryo, then the whole embryo was run on the 2 NGS-based platforms, the euploid result could occur. I've included a few screenshots here of the CNV charts generated by our experiment to demonstrate the great quality of sample data we achieved and how confidently the discrepant samples were seen to be euploid via the 2 NGS-based platforms. They showed absolutely no trace of the abnormalities previously predicted by SNP array. Here we see 2 sets of embryo trophectoderm pairs run on both ReproSeq and VeriSeq, with the gray and yellow charts representing ReproSeq and the green charts representing VeriSeq. So if Mosaicism cannot account for the discrepancy between the SNP-based assay and the results seen by both NGS-based assays, what can? In my opinion, the most plausible explanation is that sample noise was erroneously interpreted as aneuploidy during SNP analysis. This is a critical reason why we must have good tools to properly assess the quality of the data generated by a given sample, and why relying on AI software to distinguish sample noise from true abnormalities is a bad idea. Here we see two examples of samples that did not pass quality control in our lab. Even to the untrained eye, these copy number charts look drastically different than the nice clean ones we saw on previous slides. When there is this much sample noise, one cannot possibly provide accurate results. The proper call for such samples is no call, and the embryo's ploidy status must be treated as unknown. I'm a firm believer that it is better to have more no calls than to risk discarding potentially euploid embryos. Next, we wanted to test the reliability and precision of the ReproSeq assay. First, all samples from the resolution study were run a second time to ensure no call changes were seen. None were. Then we filled an entire run using only one sample 24 times. This sample had a known 40% Mosaic trisomy X karyotype and was used to check for consistency in reads per sample, as well as percent mosaicism detected by the ReproSeq assay. We averaged over 145,000 reads per sample after filtering, which was well above the manufacturer's suggested cutoff of 100,000. Additionally, we saw all samples stay fairly close to the expected 40% Mosaic value with a mean level of 39%. This entire experiment was then repeated by a second technician to verify reproducibility of these numbers in a new set of hands. Overall, the coefficient of variation for reads after filtering was less than 10% for both technicians, indicating great evenness of sample representation across the ReproSeq sequencing chip. With regards to mosaicism, we saw plus or minus 10% from the expected value of 40%. These numbers are spot on with what I have seen over the years with the VeriSeq assay through thousands of samples tested, and both represent what I believe to be the current best available precision for a commercial Pgta platform. However, it also represents one of the reasons why I do not think it is a good idea to stratify mosaicism into low-level and high-level groupings on reports. As you can see, even under unrealistically ideal circumstances, the exact same sample could show up as low-level 30% Mosaic in one instance and high-level 50% Mosaic in another, a difference of 20%, even within the very same sequencing run. It is better to report only what is seen rather than to attach a conclusion to the embryo, which may be untrue. I'd like to take a moment here to discuss reads per sample, because I have heard concerns in the field pertaining to VeriSeq achieving far more reads per sample than ReproSeq. But looking at only reads per sample is not a fair comparison. First, on average, a much larger percent of reads are usable with ReproSeq compared to VeriSeq, 85% versus 50%. But more importantly, the read length is much longer with ReproSeq, 110 base pairs per read compared to 36 base pairs with VeriSeq. That means our average of 145,000 utilized reads per sample equates to 16 million total base pairs with ReproSeq, and 500,000 reads utilized with VeriSeq equates to 18 million base pairs, which is a far cry from the difference implied by someone stating that VeriSeq generates 1 million reads per sample compared to 145,000 with ReproSeq. Nonetheless, it's true that VeriSeq can generate slightly more data per sample than ReproSeq, and that is precisely why we ran our experiment. I needed to know empirically, do their differences result in different outcomes? And all of our data said no. Both assays yielded identical results on all samples tested. So having found the resolution and accuracy to be the same between ReproSeq and VeriSeq, we then took a closer look at software and protocol differences. With regards to data analysis software, we found that the Ion Reporter software used by ReproSeq did offer some exciting new features compared to the now aging BlueFuse software used by VeriSeq. Ion Reporter can automatically flag segmental and mosaic aneuploidy events, whereas BlueFuse can only make whole chromosome calls. With BlueFuse, it is still possible to make segmental and mosaic calls based on the CNV charts, but the software cannot flag them automatically. I say flag because I do not believe in setting power thresholds for mosaicism for a whole host of reasons. But long story short, I think every sample called aneuploid deserves the attention of an expert analyst to properly assess the validity of the call. I compare it to the reCAPTCHA security features we encounter online, utilized by websites to determine whether a user is human. If a computer cannot be relied upon to do as simple a task as determining which of the tiles in this picture contain street signs, how can we possibly rely on AI to sort through millions of genomic data points to tell us which Pgt samples are mosaic? And the same goes for segmental deletions and duplications. The human mind is still the best image recognition device in the world. It's not to say I don't want the software to flag possible mosaicism. That's really important. I just believe that manual review is still essential at this point in time. The Ion Reporter software also includes coverage maps for the assessment of data quality, and it allows for the use of custom baselines should we ever need to use a different amplification method, say for example when using multiple displacement amplification for targeting the difficult to amplify single gene disorder for concurrent PGTM testing. Both the ReproSeq and VeriSeq protocols can be done inside 12 hours, but we find more practically they can be split over 2 workdays. The ReproSeq protocol can utilize 3 different size sequencing chips, allowing for run sizes of 16, 24, and 96 samples, while the VeriSeq protocol has only 1 chip size intended to run 24 samples at a time. Whole genome amplification for both ReproSeq and VeriSeq use the same chemistry, and the WGA workflow is essentially identical with the exception of adding barcode indices in the 3rd stage of the WGA process with ReproSeq, which allows for pooling of the samples prior to template preparation. The biggest difference between the 2 assays is ReproSeq's plug-and-play automation of the lengthy and sensitive template preparation chemistry needed prior to sequencing. This is accomplished via Thermo's Ion Chef robot. It essentially frees up 5 hours of tech time compared to the manual VeriSeq protocol. Additionally, the Ion Chef gives you the great flexibility in that it allows you to dictate precisely when you would like the robot to finish. You can tell it to be done in as fast as 5 hours, regardless of the run size, or you can tell it to finish up at, say, 9 a.m. the following morning, so the sequencing chip is ready to load onto the sequencer at the beginning of the next workday. Here you can see the overall protocol length is very similar for these 2 assays at the 24 sample per run level. However, ReproSeq does have a scaling advantage as VeriSeq is limited to a capacity of 24 samples. With VeriSeq, each additional set of 24 samples will add 3 hours of sequencer time, plus at least an hour of extra prep time, and that's assuming all samples are prepped simultaneously. If prepped separately, the total time doubles. With regards to hands-on times reported by manufacturers, you must be careful to note the duration and frequency of breaks between steps. For example, though the template preparation portion of the VeriSeq protocol requires only 90 minutes of direct hands-on time, in practice, our technicians are tied up for about 5 hours, as the longest break in the protocol is just 35 minutes. I think the more practical metric here is uninterrupted hands-off time. Both assays afford the user about an hour of free time during the whole genome amplification process, but the Ion Chef robot frees up an extra 4.5 hours of usable tech time compared to the manual VeriSeq library prep. And finally, cost. Our lab saw only a modest reduction in kit costs for ReproSeq compared to VeriSeq, about 15% at the 24 sample per run level. However, the ReproSeq workflow also offers some less apparent but very substantial benefits. First, because the ReproSeq samples are barcoded during the whole genome amplification stage, they can be pooled before downstream processing. This makes the ReproSeq workflow much simpler and results in far less labware being used as opposed to the VeriSeq protocol, where samples cannot be pooled until right before loading the sequencer. After whole genome amplification, the ReproSeq protocol essentially uses the same amount of reagents and plasticware whether processing 1 or 96 samples. With 96 samples in the VeriSeq workflow, you use at least 96 times more tips, plate wells, purification beads, etc. Does this drastically alter the cost per sample? No, but it easily tacks on a couple $100 per run, which adds up over time. Additionally, unlike VeriSeq, the ReproSeq assay does not utilize hazardous reagents, which means you do not need to invest in a fume hood, and you do not need to pay for expensive hazardous waste disposal. But the feature that grabbed my attention the most with ReproSeq is how much simpler and more reliable the workflow is using their Ion Chef robot to fully automate template preparation. Copy number assays inherently involve sensitive reactions that require experienced and expertly trained laboratory technicians to achieve high-quality results consistently. Being able to automate going from pool to ready-to-sequence chip on the Ion Chef allows us to run more samples with fewer expert technicians, while simultaneously removing some of the human imprecision from the process. In summary, we were quite impressed with the ReproSeq PGS platform. Empirically, we could find nothing that VeriSeq could detect that ReproSeq could not, and subjectively, we found the ReproSeq assay much easier to use. The Ion Chef makes processing samples much simpler, and data interpretation is also easier on the more powerful Ion Reporter software. Combined with the reduction in costs, we have found ReproSeq to currently be the best assay for our lab, and is now what we use on a daily basis at NextGen MDX. I'd like to end by saying that I love that there is an exemplary newcomer in the Pgta space. In my opinion, VeriSeq PGS had no equivalent competition for nearly a decade. And likely because of that, I saw next to no innovation on their product over that stretch of time. Competition fosters innovation, and the team at Thermo Fisher did a great job listening to the needs of those in the Pgta field. I can personally vouch for the fact that they are regularly reaching out to myself and other Pgt experts for guidance on how to make the next version even better. I'm glad to see this because as much as I have enjoyed working with their team, my first and foremost goal will always be quality. So keep up the good work, Thermo. And with that, I'll hand it off to Carolyn Mack, Senior Market Development Manager of Reproductive Health at Thermo Fisher Scientific.
Thank you very much, Adam, for a very informative talk. Like Adam said, I'm Caroline Mack, and I'd like to take this opportunity to share a little bit more about our solutions that can help meet your goals. So let's start from the top. Again, as you probably already know, reproductive health has a very broad portfolio. But if we look at this life cycle, starting from the left with our carrier screening portfolio, we have our microarray product called Carrier Scan, but then our newcomer is Carrier Seq, and that is done on our NGS platform, and it screens for 420 genes. Moving on to the right, we have our pre-implantation genetic testing solutions, the ReproSeq that you have just heard about. And recently we added PGD-Seq as well. That is our PGT-M solution. If you go down, continuing down to the right and the left, there is a prenatal and postnatal solution as well. And that is our high-density SNP microarray. And at the bottom, we have newborn screening, which we will have offerings soon, so stay tuned for that. Again, all these innovations and solutions is really to help future generations be healthier and also to accelerate growth with a comprehensive menu of reproductive health solutions for you. And again, with our Follow the Sun model, we provide the top-notch customer service to keep your lab running day and night. Now I want to move on to our questions and answers section. So I'm going to turn it over to Michelle, our moderator.
Thank you very much, Adam and Caroline, for your presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We will answer as many questions as we have time for today. All right, let's go ahead and get started. Our first question is, how long did it take to compare the Pgta platforms?
So that took us about 3 months to do, but most of that time was spent trying to find a partner lab and obtain the samples to test for the portion of the study in which we were comparing embryos that had been previously tested by SNP array to the NGS-based platforms. I think if it was easier for your lab to obtain samples, say you're an IVF lab, you could do it much faster than that. The testing itself could be done in a couple of weeks, I think.
Thanks, Adam. Okay, next question. What were the two biggest deciding factors for a transition to Retrofit?
So first and foremost, it's quality. We had to be absolutely certain that we weren't giving up anything in a move to a new Pgta assay. We wanted to know, it's a thing of trust but verify is the term I'm looking for. We wanted to look at the marketing materials and then put it to the test. Say empirically, it really doesn't matter what the manufacturers are saying. We want to know, does a difference in the platform result in a difference in the outcome? And we saw it didn't. So that was the first thing that we had to verify. And once we knew that that was true, then we placed more importance on other ancillary things like capital costs, cost of the assay, ease of use in the lab, tech time, all those things then came into play, and they were all in favor of Retrofit. So that's why we ultimately made the decision to move to their assay.
Great, thank you. Now, what recommendations can you make to anyone who wants to test the leading Pgta platforms in their own lab?
Again, I would say you really want to get your hands on it and try it out for yourself. I know that can be easier said than done depending on your particular situation. If you don't have the sequencing equipment in your lab, work with your local representatives, see if demos are a possibility, if you know another lab that has the equipment that you can run it on. But really, you need to have firsthand experience to know if it's the right choice for your lab.
Thank you so much. And again, to the audience, any questions that we did not have time for today will be addressed by our speakers via the contact information that you provided at the time of registration. So thank you so much, Adam and Caroline, for your time today. Adam, do you have any final comments for our audience before we go?
No, I would just like to thank you for having me and thank everyone for listening.
Thank you again, Adam, for your time today and for your research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care. Goodbye.